We have long viewed the aging process as an inevitable clock, a slow and steady progression toward biological failure. For a therapy to reach patients, the FDA must approve it for a specific indication, a legally defined disease or medical condition that the drug is intended to treat. Researchers are now lobbying to recognize aging as an indication. They argue that the decay we associate with growing older should be classified as a treatable pathology rather than an unchangeable fact of life. This view shifts the focus from time itself to biology. Tissue deterioration is an active process, a series of programmed cellular events, rather than the simple wearing down of an organic machine. This circular diagram maps the hallmarks of aging. These 9 distinct biological drivers are specific microscopic cellular glitches. Looking at categories like genomic instability or cellular senescence, we see human decline is dictated by mechanisms causing the body to fail. Identifying these hallmarks changes how we view the finality of aging. If biological mechanisms drive deterioration, then those mechanisms can be intercepted and treated. Consider telomere shortening. Every time a cell divides, the protective caps of its chromosomes get shorter. Eventually, these caps are too short to protect the genetic code. The strands fray and unravel, leading to severe instability that halts the cell's ability to replicate. This leads to cellular senescence. As shown in the top row of this chart, damaged cells, highlighted in blue, stop dividing but fail to undergo natural cell death. Instead, they linger. In the pathogenic outcome row, we see how these lingering cells damage healthy neighbors by constantly secreting inflammatory factors. To fight this, researchers are testing a class of drugs called senolytics. Experimental compounds like dasatinib and quercetin work by selectively identifying and clearing out these lingering cells to prevent them from damaging healthy tissue. While human results are still pending, these senolytics prove we can develop precise interventions for one of the most destructive hallmarks of aging. Our genetic software also degrades. This hourglass graphic shows how processes like DNA methylation alter which genes are active, a phenomenon known as epigenetic drift. We can now track this degradation using epigenetic clocks. These models measure DNA damage against our body's adaptations to calculate our true biological age. Measuring this clock allows us to test reboot drugs like rapamycin. By inhibiting a protein complex called mTOR, rapamycin triggers cellular maintenance systems usually only activated during caloric restriction. In NIH-led trials, rapamycin significantly extended mouse lifespan. Median longevity increased up to 26% in females, 23% in males. Combining rapamycin with other drugs like acarbose shows an even greater effect, with longevity gains exceeding either drug alone. However, success in mice does not always translate to humans. NAD+ precursors, for example, have faced significant setbacks. Despite boosting cellular energy in isolated tests, these compounds have failed to significantly alter the overall lifespan trajectories of mammals in clinical trials. These failures highlight a massive gap between single cells and whole organisms. Moving from mouse to human requires navigating a thicket of metabolic crosstalk and systemic feedback that a single compound cannot easily resolve. We are now attempting to bridge the gap between extending life in a petri dish and achieving those same results in a living human being. Human biology is immensely more complex than that of a laboratory mouse. Translating these therapies means identifying and managing systemic side effects that only emerge in higher-order organisms. This is why the first human trials are focusing on prevention. Instead of chasing immortality, they are testing if these therapies can stop the cardiovascular events and frailty associated with aging. The goal is a systematic extension of health span, ensuring the years we are alive are spent in a state of high physical and cognitive function. This data, gathered from mouse and human cohort studies, has identified the specific molecular pathways where our cells fail. This moves aging from a philosophical inevitability into a measurable biological target. We are entering a phase where the drivers of decay are treated like any other clinical pathology.